Biochemistry 401, Lecture 35. Today we're going to talk about replication. We're going to talk about replication in bacterial and eukaryotic systems, and it's important to keep these two straight. We're going to discuss key enzymes and proteins that are involved in replication and also the regulation of this process. We're then going to talk about topoisomerase 1 and 2 specifically. So let's get started. Hi, today we're going to talk about replication. We're going to talk about replication in two model systems, E. coli for prokaryotes and humans for eukaryotes. Replication is a little bit different in each of these systems, so it's important to get this straight. So as with any system, it's important to think about things in a big picture perspective. And for replication, we want to concentrate on these things. First of all, what is happening? When does this happen? How does it begin? How is this beginning regulated? What enzymes are doing this? Are there any accessory proteins that help these enzymes to know where to start and be able to continue? What is the directionality of these enzymes? Do they go 5' to 3', 3' to 5'? It's important to know this. So, with this in mind, let's get started. Replication is DNA synthesis in preparation for cell division. Replication is semi-conservative. This means that the parent DNA separates and a daughter strand is made from each parental template so that the parent cell keeps a parental strand and a new daughter strand, and the daughter cell gets a parental strand and a new daughter strand as well. The process of replication in bacteria and in humans requires similar things. In each, replication requires a DNA template, so in this case it's the parental strand, and it requires ribonucleotides to make RNA primers, because first you must make RNA before you can make DNA. Deoxyribonucleotides for DNA synthesis are also needed. And finally, polymerase enzymes, accessory enzymes, and proteins are also required. So those are the things that are similar, but what sorts of things are different? In bacteria, there is generally one circular chromosome. In E. coli, this is true, and there are about 4.6 million base pairs on that chromosome. Each circular chromosome has one replication start site, and the cell division is based on cell size. This is the relative surface area to volume ratio. Each different bacterium has its own particular optimal size, and once that size is surpassed, it's time to divide. In eukaryotes, for instance in humans, the chromosomes are linear. There are 46 linear chromosomes in humans and approximately 6 billion base pairs. That's a lot more DNA that must be copied during replication. And as you'd expect, there are multiple replication start sites to get this big job done quickly. Cell division is highly regulated and differs with respect to cell type, developmental state, intracellular signals, and extracellular signals. Let's talk about replication in E. coli first, because this is a little bit easier to understand. First of all, where does replication start on the chromosome? And what sort of enzymes are responsible for this beginning? In E. coli, the initiation of replication occurs at a place called the origin of replication. That makes sense, doesn't it? It's abbreviated ORI, and in E. coli, it's ORI-C. The ORI-C is a DNA sequence in E. coli that consists of two main regions. The first is an AT-rich region. The AT repeats represent a site at which the DNA is easy to melt because there are only two hydrogen bonds between AT base pairs, and so it's relatively easy to melt. The second portion is the DNA A binding site. This is a site to which DNA A binds. This DNA A recognizes the sequence and it binds in these specific spots and uses ATP hydrolysis to power the separation of the DNA strands at the Ori locus. So now that the DNA is separated, it's important to make sure that this DNA does not reanneal or form higher order structures like stem loops or things like that. We don't want our DNA to get bound up in knots. And so we're going to cover this DNA with something called single strand binding protein, SSB, that coats the DNA and prevents it from reannealing or forming higher order structures. The next thing that happens is DNA A is going to help recruit DNA C and DNA B. So it's important to know your ABCs of bacterial replication. DNA A binds first. DNA B is loaded onto the DNA by DNA C. What does this mean? DNA B is a helicase that consists of 6 individual subunits that comprise a ring-like structure. DNA C opens this up and helps to place this DNA B on the chromosome at the start site of replication. So DNA A binds first, helps to separate the strands. DNA C binds and helps to load DNA B, the helicase, onto the DNA. So DNA B is associated with the primase. This primase is called DNA G in E. coli. And this primase is going to synthesize RNA primers that help get replication going. Because remember, DNA synthesis cannot happen without a primer first. And in this case, we're going to use an RNA primer. RNA first, then DNA. So DNA G is bound to DNA B, and DNA G is the primase, and it synthesizes a short strand of RNA as a primer. And then DNA synthesis itself can get going. As we can see from this illustration, the DNA is going to be synthesized in 2 directions. There are actually 2 chromosomes made up of DNA B and DNA G that are found on this chromosome. There's one going to the left and one going to the right. And so we're going to be replicating in 2 directions at once. Now the place at which this DNA is separated, okay, on either side, is called a replication fork. And it's called a replication fork because it looks like a fork in the road, right? And so we're going to have one to the left and one to the right. And right now, we have the primosome bound DNA B and DNA G. This is going to help to recruit the main replicative enzyme, which is polymerase 3. And this enzyme is part of what's called a replisome. The replisome itself consists of 3 main parts. We have a beta-2 clamp that helps to hold the polymerase enzyme on the DNA strand. And we also have the polymerase itself, of course. So Pol3 core polymerase contains 3 portions. An alpha, which is the actual catalytic portion. The theta, which frankly we really don't know what it does. And the epsilon. The epsilon actually is the exonuclease capability. So if this polymerase makes a mistake, the epsilon can take off that nucleotide and give the polymerase another chance to get the right one there. But we have to have something that's going to open and close the beta-2 clamp to get it onto the DNA and to release it. And this is called the clamp loader. Makes sense, huh? So each replisome is going to have a clamp loader, beta-2 clamp, and polymerase, which is Pol3. And one's going to go to the left and one's going to go to the right. And each one of these replisomes is going to synthesize 2 strands of DNA at the same time, the top strand and the bottom strand. This brings up a problem. We know that DNA is synthesized 5' to 3', and the DNA template is read 3' to 5'. But remember the convention? The top strand is 5' to 3', and the bottom strand is 3' to 5'. So if we separate these two, one's going to be going in the right direction to be used as a template and read 3' to 5', and the other one's going to be going backwards. So we have two replication forks, one going this way, one going that way. It's going to synthesize two strands of DNA. One's going in the right direction. It can be read 3' to 5', no problem, and the other one can't. And how do we fix this? Well, we have to keep the forks going in the proper direction. We're going to reverse one of the strands in each of these forks and synthesize them in little tiny bits called Okazaki fragments so that both strands can be copied at the same time. Let's take a closer look at that because it's a little bit complicated. So here in this slide, we can see a replication fork that's progressing in the rightward direction. And on the bottom, we have the lagging strand, and on the top, we have the leading strand. Now, the leading strand has already had its RNA primer synthesized, and so it's just chugging along no problem. The clamp has been loaded onto that strand, the beta-2 clamp, and the core polymerase consisting of alpha, theta, and epsilon are there. So here we have the top strand. It has the beta-2 clamp on the DNA, and the polymerase is attached also, and it's going in the rightward direction, no problem. It's reading the DNA 3' to 5'. It's synthesizing the daughter strand DNA 5' to 3'. No problem for the leading strand. The lagging strand, that's the problem. It's turned backwards so that the polymerase can synthesize 5' to 3', reading the DNA strand 3' to 5' in short Okazaki fragments. Now for each fragment we have to synthesize an RNA. So what does this mean? The DNA B is the helicase, right? And that's going along, opening up the replication fork, and the DNA G is attached. And it starts to synthesize the RNA primer. And then the polymerase can bind to the RNA, to where that RNA is, and can start synthesizing the short stretch of DNA. Once this DNA gets to a specific length, the beta-2 clamp will be released, and therefore the polymerase will also be released. And the clamp loader will load another beta-2 clamp a little further down on the DNA, and another polymerase will bind to make another Okazaki fragment. We can't have that RNA stay there because we know that RNA is less stable than DNA. We don't want to have RNA inside of our genome. We want it all DNA all the way through. And so there's another enzyme that's going to be employed, and this is called DNA Pol I. Now, Pol I has the capability of taking nucleotides out, going 5' to 3'. So it acts almost like a snowplow to remove RNA in the Okazaki fragments and replace that RNA with DNA. So in E. coli, DNA Pol I removes the RNA primer and synthesizes DNA to replace it. But the thing is, we still are going to have breaks in the DNA now, where we have stopped synthesizing the DNA and where the next Okazaki fragment DNA is. So we have to fix that. And DNA ligase uses ATP to ligate that DNA and seal the break. So we need the following enzymes for replication in E. coli. First, we need DNA A in order to separate the strands. We need DNA C to recruit DNA B. DNA B is the helicase. DNA G is the primase. The main polymerase is Pol III. It's going to synthesize the leading strand and the lagging strand. The lagging strand is synthesized in short fragments called Okazaki fragments. Once those are made, Pol I comes along and removes the RNA from those Okazaki fragments and fills those in with DNA complementary to the template. And then DNA ligase seals the break to make a continuous strand of DNA on the lagging strand too. Finally, both sides of the strands are synthesized so we can have one parent strand and one daughter strand remain with the parent cell and one parent strand and daughter strand going to the daughter cell. So each bacterial cell will have its own genome, consisting of one parental strand and one daughter strand. I hope that's clear. So what do you need to know? You need to know what DNA A, DNA C, DNA B, DNA G do. You need to know some of the things that comprise the replosome, but only these. You need to know the beta-2 sliding clamp. You need to know the core polymerase, which consists of alpha, theta, and epsilon. You need to know what alpha and epsilon do. You also need to know that there is a clamp loader, but you don't need to know the subunits in the clamp loader. Pol I comes along and removes the RNA from those Okazaki fragments and fills those in with DNA. And then DNA ligase seals the break. I hope this helps. This also might be helpful. There are two links for DNA synthesis that might be helpful to you. In eukaryotes, it's a lot more complex. It has to do with the tissue type, it has to do with the developmental stage, signals inside the cell and outside the cell that say it's time to divide. And only particular cells have the ability to divide. Those that don't have the ability to divide have exited the cell cycle and are now at something that's called G0. This is the non-dividing stage. For a cell that's going to divide, there are four main stages. There's G1, which is the growth phase. There's S for synthesis of DNA and other things that are going to be necessary for division. And G2, where the cell is growing again, but is also checking and making sure that everything is just right, that the DNA has been synthesized correctly, and if it hasn't, that it's been repaired. And then mitosis begins. In eukaryotes, replication is complex and highly regulated. There are several linear chromosomes, and on each chromosome there are many origins of replication. In fact, there are 10,000 to 100,000 origins of replication in the human genome, and each one is used once per cell cycle. And so in order for replication to occur correctly in humans, each of these origins of replication must all start at the same time and they must all finish before G2 phase. So how does this happen? Well, the ORC, the origin of replication complex, is a complex of 6 proteins that binds to the origin of replication in a similar function to DNA A, and using ATP hydrolysis unwinds the DNA and then recruits proteins called licensing factors. These are similar to DNA C and DNA B in a way. These licensing factors include CDC6 and CTD1. These are similar in function to DNA C in a way, although they're much more highly regulated than DNA C is. They are recruited by ORC and they load the helicase MCM27 onto the DNA. Now, MCM27 is similar in function to the E. coli helicase DNA B. It's called the mini chromosome maintenance protein. Do you need to know that? Nope, you just need to remember MCM27. Again, this is a helicase that helps to unwind DNA at the replication fork. Similar to E. coli, there will be 2 MCMs that are going to be loaded onto each origin of replication, one going to the left and one going to the right. So there will be several of these helicases loaded onto the chromosome prior to S phase. And so during G1, ORC binds, it unwinds the DNA a little bit, recruits CDC6 and CTD1, and MCM27 helicase is loaded and ready for the S phase to start. During S phase, DNA polymerase A primase complex synthesizes RNA primers on both the leading strand and Okazaki fragments. Now, this is similar in a way to DNA G, right? But DNA Pol A primase complex also includes Pol I functions. So this polymerase can make these RNA primers, but this also can synthesize DNA as well. And so in a way, it's also similar to DNA Pol I in E. coli. Another thing that's different also is that instead of just having one main polymerase, like the DNA Pol III in E. coli, in humans there are two, DNA Pol delta and DNA Pol epsilon. DNA Pol delta does the difficult work, D for delta, D for difficult, of synthesizing the lagging strand, whereas DNA Pol epsilon, E for epsilon and E for easy, synthesizes the easier leading strand. Again, these are similar in function to Pol III. These polymerases are held in place by a sliding clamp that has similar function to beta-2 sliding clamp in E. coli. This is called the proliferating cell nuclear antigen, or pCNA, and you're only required to remember pCNA. And again, this holds the DNA polymerase on the DNA, similar in function to the beta-2 sliding clamp that we saw in E. coli. It's important to be able to figure out which of these eukaryotic proteins and enzymes are similar to those that we've already seen in E. coli. You'll be expected to know this. Another thing that's different in eukaryotes is that they have linear DNA. And so at the ends of this DNA are something called telomeres. And this is a portion of the DNA that consists of repeats that are folded on one another to protect the ends of the DNA so that during replication, these ends don't get shorter and shorter and shorter and compromise the actual coding regions on the DNA. So telomeres are at the end, and during replication, those have to be replicated also. And there's an enzyme that's called telomerase that does that. Telomerase is a reverse transcriptase that has an RNA that's used as a template for the synthesis of DNA. So telomerase carries along its own template. It binds to a portion of the telomere, and it then synthesizes DNA, complementary to the RNA template that the telomerase carries around with it. So the telomerase finds the telomere, it binds, and it uses its own RNA template and the reverse transcriptase to make DNA to extend the telomere. And then once the telomere is formed, it folds on itself to make the end of the chromosome. Now the thing is, with all of this unwinding, it's really easy for the DNA to become overly coiled. And so it's important to make sure that we can take care of the supercoiling so it doesn't break the DNA. There are things called topoisomerases that break either one strand or two strands of DNA to release this tension to become less supercoiled. There are some topoisomerases that put supercoils in, and there are other ones that take them out. So let's just say that this is DNA, and it has a certain number of times that one strand wraps around the other. This wrapping of one strand around the other is just because of the nature of the DNA itself, the length of the DNA, and the structure of the nucleotides that make up the DNA. It tends to want to wind around one strand winding around the other every 10.4 base pairs. So there are 10.4 base pairs for every turn of the DNA. And again, this is just because of the structure of the DNA itself. If you were to know the length of your DNA, you could find out how many turns there are. These turns are called the linking number. So for something that is 104 base pairs, it's going to have a linking number of 10. The thing is, is that you can change the linking number, but if you change the linking number, you're going to introduce supercoiling, and that's what happens like this. See how this DNA has turned on itself so that the double strands are actually crossing each other like this? This is a supercoil. This crossing over of one double strand over another is called a writhe. And the number of times that the DNA is turning around itself now, because it's just allowed to do whatever it wants, I'm not holding it straight, okay? I'm letting it do whatever it wants. This is called the twist. So the linking number and the twist are different in that with the linking number, oops, I broke my DNA. Topoisomerase in action. Okay, sorry about that. So here's the linking number. This is linking number, and it's caused just by the one strand going around another, right? This is an effect of the number of nucleotides, number of base pairs in the DNA, and the structure of the nucleotides themselves. 10.4 per turn, linking number. This is holding it in a single plane. I'm holding this DNA in a single plane. I'm not letting it do what it wants to do. It wants to turn around and form writhes and stuff. So now I'm going to let go. It's formed writhes, and what's left, these little turns that are going around each other, are called twists. Twists plus writhes always equal the linking number. Again, twist plus writhe equals linking number. So for instance, there are, oh Lord, 1, 2, 3, 4 writhes in this. Okay, so if I knew what the linking number was, I would just subtract the writhes from the linking number, and I'd know how many twists are in there right now. So, but you have to know whether it's going to the right or going to the left. So that's a little bit different. Let's look at this a little more closely. We'll do a couple of these together. You have to know this. So you have a DNA that is 104 base pairs. It has a linking number of 10. If there are 2 negative writhes, that means you're going to have a twist of 10 plus 2. So that's twist of 12. If the DNA is supercoiled in the other direction, you're going to have, and you have 2, it will be 10 minus 2 equals 8, a twist of 8. So do you have to be able to figure out whether it's a negative or a positive supercoil? I'll tell you that. If I give you a question like this, I will tell you exactly whether it's a negative supercoil or a writhe of -2 or +2 or whatever. So you're not going to have to figure that out on your own. Promise. That's too tough. Okay, so we talked about topoisomerases. There are 2 major classes, and lucky for us, when they named these things, they did it the right way. Topoisomerase 1 cuts the DNA once in one strand, and it uses the tension that's already in the DNA to unwind the DNA and change the linking number by 1. So either adding a linking number or subtracting a linking number. And there are different topoisomerase 1s that either add or subtract a linking number. So no ATP is needed for topoisomerase 1. Topoisomerase 2 is another subject. Topoisomerase 2 requires 2 ATP, makes double strand break, so it cuts 2 strands of DNA, and it changes the linking number by 2. Topoisomerase 2, 2 ATP, 2 strands of DNA are broken, and linking number is changed by 2. Let's look at how these topoisomerases work. Topoisomerase 1 uses the catalytic tyrosine to break the phosphodiester backbone, and it does so by transesterification, So it just replaces one ester bond, that's the phosphodiester bond, with another one. This is an ester bond from the backbone to the tyrosine. So the tyrosine in the enzyme breaks the phosphodiester bond and holds one strand, and the other one unwinds by one. And this is topoisomerase 1, uses transesterification with the catalytic tyrosine and lets the other strand unwind, changing the linking number by 1. Once this DNA has unwound, the tyrosine releases the DNA and the phosphodiester bond is regained to seal the break in the DNA backbone. So for topoisomerase 2, I'm going to get this out again. Here's my DNA. This is the place where it's going to be broken. So this DNA is called the G segment. It's going to enter into the gate. The gate is going to bind. It's going to break this DNA. And now there is a double-stranded break in the G segment. Now, I need another arm. I need another hand. Okay, now that we have the G segment that's broken, the T segment is going to go through. I hope you got that. The T segment has gone through the break, and now all that has to happen is the DNA breaks are repaired. ATP is hydrolyzed to release the DNA, and it all starts again. 2 strands of DNA are broken. Linking numbers change by 2. 2 ATPs are used. This marks the end of Lecture 35. Have a great day.